but it should be easier for you guys to write stuff down. Okay, so the main tools that we use to non-invasively, or in some cases actually invasively, measure muscle function. Two of the primary ones are electromyography and mechanomyography. We're going to talk about EMG first. EMG came around a little bit earlier than MMG, and it's more commonly utilized, although the exact inferences you can get from EMG and what's commonly accepted has definitely changed in the last 10 years. So interpretation is a little bit different than it originally was. But what EMG is going to allow us to partially do or estimate is a measure of electrical activity of the activated muscle or muscle fibers. The amount of muscle and which specific fibers you're actually measuring is dependent upon the type of EMG you would be using to do this kind of data collection. But really, it's just a measure of activation. So we're going to do some review with action about what action potentials are and voltage and ligand-gated channels in our next lecture. But just keep this in mind as you're learning about EMG, is that that's what it's looking at. It's looking at action potentials moving down the muscle fiber either itself or muscle fibers as a bundle and taking that electrical activity, giving us then a screen interpretation of that activity that we can then analyze and try to get some inferences on what's going on within the working muscle, which is pretty cool. So when you are getting ready to do some type of muscle action, usually a voluntary muscle action, you are going to send a voluntary signal from your central nervous system down the spinal cord. That's going to attach to a sensory efferent fiber, and that efferent fiber will then send a signal down the motor or the axon of your motor neuron using, usually in that case, a voltage-gated channel. So voltage-gated just means there's some kind of electrical potential change occurring. Voltage-gated potentials, as we'll discuss in our next section, only occur down the axon. Right? So down the axon of that nerve fiber, that's where you're going to see these voltage-gated channels. And that change in electrical potential occurring down the muscle fiber is what your EMG or surface sensors are picking up. Now, what it picks up is really dependent upon where you place those sensors. If you place those sensors directly over what we call an innervation zone, that means the point at which that nerve and that motor unit are coming together, so the nerve is innervating that muscle fiber, we'll actually see a decrease or a cancellation of our EMG signal, so we won't pick up anything. If we move our exterior surface electrodes down the muscle belly along the skin, then we'll start picking up potential, but that potential will decrease as we move further and further away from the innervation zone. So at the innervation zone, we have a zone of cancellation, so a zero signal. Just on the other side, say on the other side of the innervation zone, you're going to get a positive change, so we'll be able to pick up some electrical potential. But the further away you go on that muscle fiber, away from the innervation zone, away from the source of that electrical change, the lower your EMG signal is going to be. So when we start placing these sensors, there are specific criteria and recommendations for these sensor placement to avoid innervation zones and to ensure that we get the best possible estimate of that action potential or that change in electrical potential down the muscle fiber. So because EMG is, and this is the original interpretation, was that EMG measures the membrane potential. That's what the first inventors of it said it did. But in order to actually do that, if you remember that a membrane potential is generated, and if you remember, think back to basic physiology, which we'll rehash again in our next section, that you have your membrane, you have your axon, and a potential is created because of a difference in voltage from the outside and the inside, right? So you have positive The outside negative, as you become more electronegative, you get closer to reaching your action potential. So in order to actually measure that, theoretically, you would need an electrode or a sensor on the outside of the fiber and the inside of the fiber, so you can actually detect that electrical change. Surface EMG doesn't do that. Even intramuscular EMG doesn't do that because it doesn't have that placement both outside and inside the fiber. It's either on top of the skin, or in the intramuscular EMG, it's located under the skin but not within the fiber itself. So the interpretation of EMG is slightly changed so that it's no longer saying, we no longer say that we can detect the actual membrane potential changes, but we're looking at the general field of potentials associated with current underlying the muscle. So we have a current going through specific muscle fibers. We're going to estimate in general what that current is going to be. It's not exact because, again, we are only on the outside of that muscle fiber. And in the case of surface EMG, we're on top of the skin. So there's additional layers like subcutaneous fat and connective tissue that we're dealing with in our interpretation of that signal. So we're going to use these extracellular field potentials to then estimate the membrane potential. And based on that, we can give some kind of estimation on muscle activation or the overall stimulus that these muscle fibers are receiving from the central nervous system. So what is EMG signals, or what do they look like? There's different filtration processes that are used to help create as clear an estimated signal as possible. But in our lab, you're going to see something like this. So we have a raw EMG signal here in green. Each of these bursts represent a contraction. So this portion here, you have sensors, and this is an example. These are surface wireless sensors. This is an intramuscular sensor, so it's usually be more precise, but you're going to get a smaller estimate of action potentials. It's more invasive. And there's less activities you can do with the intramuscular sensor. So usually these are limited to like MVCs, maximal voluntary contractions, sitting, or isometric contractions of the upper body. Wireless sensors, and even some wired sensor systems, we're going to use like a pseudo wireless system for our lab, allow for more movement and flexibility. But so here we have our raw signal. We're going to place, in terms of a surface EMG, we're going to use what's called a bipolar arrangement. So for every signal you see here, that's picked up by two different sensors. So if you look here in this photo, you can kind of see that there's two sensors around the same, or these are actually two different muscles, but they have, this is in a biomechanics lab, so they have their light sensor to pick up the movement or placement of that joint, and then you have these different EMG sensors. These are actually two different sensors on the same electrode. We're going to use a pseudo wireless, which we're going to have separate, they look like stickers, or like an ECG electrode. And we're going to place those right on top of the muscle belly that we're interested in. And these two signals are going to pick up the action potentials coming towards and away from that innervation zone. So we're going to use a differentiated signal, and the result of that differentiated signal is going to show up here on our screen. So we place our electrodes. We're getting a little bit of a signal here. You can see like a low level contraction. When you put surface electrodes on, they have a tendency to pick up any noise. So it's really important when you're placing surface EMG electrodes to make sure the skin is really clean. So when we do our lab, I think we're going to do the upper arm. But if you have any hair, it has to be shaved at the electrode site. Sometimes if it's winter and really dry, we have to use some sandpaper to get the first layer of dead skin cells off. Alcohol, nice clean surface, because that's going to decrease any gaps between that electrode and the skin. When you first put the signal or the sensors on and you're measuring any kind of activation, we can pick up noise in hertz. And if it's 60 hertz, that represents environmental noise. So like the noise that comes off the lights, noise that can come off from pipes running underneath the building. So you always check to try to make sure that environmental noise is eliminated before you actually do a test. So once you have everything cleared out, my little sensor keep going, then you can have the subject start doing some muscle contraction. So this would be contract, relax, contract, relax, contract, relax. You'll notice, at least in this example, that we have some smaller bursts, and as time goes on, you can see that the amplitude of those bursts increases over time. Can everybody see that? So the bursts are getting a little bit darker. So the dark kind of blobs in the middle are getting more solid in color, and they're getting a little bit wider. So this signal is showing you a representation of both the amplitude and the frequency of the EMG. And those are the two different variables that we're going to measure or kind of tease apart in interpreting that estimate of the membrane potential signal. So the frequency is an estimate of how often your nervous system is sending those action potentials. And that's, you can look here. So how quickly you see those little bursts, every little line that shows up, you can see that it becomes darker over time because you're getting more and more bursts. That's usually associated with fatigue, increases in recruitment, or sustained muscle actions. You can filter the signal to eliminate any environmental noise so it gets a little bit cleaner. You can do something called rectification, which means you're going to eliminate any negative portion of the signal. So you've just displayed the positive inflections of our amplitude and frequency. We can smooth it. And then most often you're going to see EMG amplitude represented by root mean square. And root mean square is going to allow us to take basically the highest point, or the standard deviation of the power curve is what your root mean square is. And it allows us to find that highest point, which will represent the maximal amplitude of that muscle action. So this is the process that occurs. Often software will come with this built in. You just have to specify what kind of rectification or filtration you're going to use. Other times people use specialized software like LabVIEW or MATLAB and actually program in these special filters. So if you're planning to get into EMG research, it's really good to know someone in engineering because they're usually really great at MATLAB and LabVIEW. One interesting thing also I want to point out before we kind of skip on is that if you look at the difference between these, so we have surface EMG and intramuscular, can anyone think of what might be different in our signal if we shifted from one to the other? So what might this be picking up if we're using surface EMG rather than the intramuscular? You can see how the measure might differ.
You say, like, as you were specifying earlier, sound external X would be associated with surface EMG and perhaps not as much with intramuscular?
You can actually still get some noise, not as much, but you can still get some noise in intramuscular because it looks like it's wireless, but there's still a wire connecting the signal, and the skin is still partially open from where that needle pokes through. So you still can get some environmental noise. But let's think about like the sensitivity of the different measures. So you have a needle that's going right into the skin, right into the muscle belly, versus something that's just sitting over the skin.
More, I guess, burst in the direct measurement.
So if talking about like quantity, you're going to probably get more muscle fibers that fall within the surface signal, right, where those sensors are, rather than the intramuscular. So the intramuscular is going to be more precise. It's going to allow you to look at much smaller groups of muscle fibers, whereas what we're looking at with that raw signal, that's hundreds of activated muscle fibers, right? Hundreds. Because it's whatever muscle fibers fall underneath where we've placed our sensor. So it's really hard to say the specific activities of, just one type of motor unit or a very specific subset of motor units, you can even pick up activity from other muscles, right? So the recommendations are to pinpoint or try to isolate muscles that you're particularly interested in. But if you're dealing with muscles that have that are more deep than others, you could theoretically be picking up some of that activity, some of those action potentials, even from the surface electrodes, because anything that falls within those or below those, the surface signals would pick up. So if you're dealing with more precise or single muscles, the intramuscular is very common in experiments that are not necessarily utilizing humans. They are used in humans, but if you see like really basic research done with like reflex properties or muscle contraction properties of like mice, rats, cats, frogs, they're usually always going to use the intramuscular EMG. It's still, you'll find it in humans, but probably not quite as common.
You could implant one inside.
An implant an EMG? I've never seen that before. I mean, they have like GPS accelerometers you can implant, but I've never seen a human get that implanted. Maybe that's the next gen performance measure, yeah.
Is there a relationship of like averaging muscular activation in the intramuscular to the surface?
People have done comparisons, if that's what you mean. Like they'll do like the same muscle actions with surface versus intramuscular.
Is there like a huge discrepancy?
You'll get different measures because it's not necessarily a global estimate like the intramuscular, and it kind of depends on what question you're trying to ask, like which one would be best. If you're looking at the individual like fiber properties of a specific muscle and maybe like a few fiber types, like just the type 1 fibers of this particular muscle, then you might want to use something that's more precise. Usually in like performance research, most people are going to utilize the surface just because you can do a wider range of activities. You're going to get similar information, just at a different scale. So this is just the more precise versus the gross measure.
Yeah.
So this is what it's actually picking up. If you look at the bipolar surface electrodes, let me try to put my mouse. So these would be our 2 electrodes, and these are going to be our muscle fibers. So we have our bipolar surface electrodes. We have our fiber. Each of these shaded areas represents the source of the action potential. So that would be our innervation zone that I mentioned, where you have a cancellation or no signal. And then we're going to see a change in voltage propagating both away from, kind of on opposite ends of that innervation zone. And that's what our bipolar electrodes are trying to detect. So using the differentiated amplifier, then we're going to get, this is what actually is being picked up, the raw EMG signal. Each one of these waves is represented here on the screen. So this would be a single potential being picked up, and it measures all of these single potentials across all the muscle fibers that fall within its measurement range, and you get a single signal like this. This in A is from a single motor unit. So this would be like an intramuscular EMG that's targeting just a single motor unit. So that'd be the nerve and all of the muscle fibers that single motor unit innervates, right? Because it's not just one fiber. Usually it's going to be multiple fibers that one nerve is innervating. Versus B, and that's going to be the action potentials of multiple fibers and many motor units. So if you're looking at the difference in what the signals might look like, on the left would be our intramuscular electrode, and on the right would be our surface EMG.
Have the slide in ours. Oh, okay.
That's why I'm waiting a little longer if people want to type. Yeah, I found this picture not long before class, and I thought it was a good example of showing the difference between the single motor unit and the many ones. So I last minute edit. Okay, everyone good? I can post the pictures if you guys would like those. So when we're analyzing the signal, calculus aside, because calculus is definitely involved in the interpretation of the EMG, that's why I said find someone in engineering who loves math, because all of those analysis softwares, they're basically it's calculus equations that deriving different areas of the curve and then calculating the different electrical effects and distance to or from the innervation zone. So you guys don't need to worry about that. We're in general going to break apart our EMG signal into two primary components. It's amplitude and it's frequency. So how large are those waveforms, and then how frequent we're getting signals being picked up from the action potentials. So amplitude reflects the magnitude of muscle activity. So you might hear it referred to as neural drive or just muscle activation, and it's just the magnitude of that muscle activation. It depends on multiple factors. Some of the biggest ones are the number of activated muscle fibers. It depends on electrode location, subcutaneous tissue, if you're using surface. There's a bunch of other ones I'll talk about at the very, maybe not very end, but in a few slides. And it's going to reflect the activation by the central nervous system. And remember, it's reflex, it's estimating, it's not a direct measurement. So all these things are just estimations of central nervous system drive or central nervous system activating those different muscle fibers. Usually this is going to be represented by the measure root mean square, which I just showed on the previous slide. That's going to be a measure of amplitude. And then frequency is related to the action potential conduction velocity. So how quickly those signals are being sent down the nerve to its attached fibers. The EMG amplitude sometimes is referred to as a measure of recruitment. So like how many muscle fibers you can recruit. That is not the correct interpretation of it. It's simply activation. So that could be because of more, your activation could go up as a function of either recruitment of more muscle fibers, more motor units, or of a faster or more frequent firing rate of those motor units. So typically we have, when we talk about neural modulation strategies, or how do we grade force if we're trying to produce more force, you can either recruit more fibers or you can recruit those fibers more frequently, right? And that's what we're going to try to use our EMG and MMG to determine which one of those is occurring. So if you're using EMG alone and you're looking at the amplitude, you can't differentiate between are you recruiting more fibers or are you recruiting those fibers more often. You have to kind of combine those together because it's just looking at overall activation of the central nervous system, not the specific strategy of activation. So this is definitely a flaw of using EMG alone. So if only EMG is used, you could say, yes, activation went up, so central nervous system or neural drives seem to go up, but you can't say why that occurred. Frequency is also kind of flawed, again, just because it's estimating the action potential conduction velocity, but it's not within the fiber measuring that. And so usually frequency will be used to estimate fiber type. So we know that larger motor units have faster action potential conduction velocities, which we'll talk about in our fiber section. So the larger the motor unit, theoretically, the faster the action potential. So if you have a higher action potential shown by EMG frequency, that could mean that larger, more type 2 motor units are contributing to that particular muscle action. So there are ways to kind of wiggle around these limitations, but in general, that's what these two things mean. Amplitude just is general or global activation, which could be a function of recruitment or firing rate. And then the EMG frequency is the quickness of that action potential conduction velocity. So the action potential can be influenced by a multitude of factors, just like EMG amplitude could. Remember, this is specifically talking about surface EMG. So if you're having the sensors on the surface of the skin, it can be affected by subcutaneous tissue. So often you will try the best you can to control for body fat if possible. So if you're doing a study looking at, and the primary outcome measure is neuromuscular function or utilizing these non-invasive tools, most often you're going to control for, a lot of studies will just use BMI just to make sure you're not getting any like gross obesity in the study. There's different results in the literature in terms of like how much body fat is actually going to significantly alter the signal where it's not usable. But usually if you're staying within the like kind of normal accepted ranges of average male, average female % body fat, then you're not going to see a huge influence from that subcutaneous fat layer on the signal. When you start getting into the overweight and the obese category, then you're going to see some differences and decreases in both the amplitude and the frequency signal just because it's further away from where it's being measured.
Would that still be an issue if you're doing like invasive? Obviously,
not as much, no. It's just going to limit the type of activity you could do. Yeah, yeah. The spatial distribution of muscle fibers. So for example, if you, and this is a review, right? You have your muscle nerve fiber. So you have your nerve, and then you have the muscle fibers. Your motor unit is every muscle fiber that motor neuron is innervating, right? So that's our motor unit. We have, if we can think of our muscle as, this might be a goofy example, but like pool noodles, right? So you have, if you go by Target or like the dollar store, they have a box of different colored pool noodles outside. They're all different colors, but they look fairly similar. Your muscle's kind of the same, right? So you have different types of muscle fibers on that spectrum that Dr. Bagley talked about. We have our type 1, we have our intermediate fibers, our FOG fibers, our FG fibers, and we have then our muscle or our nerve. The nerve is going to innervate usually all the same types of fibers. So if you have one nerve coming from your spinal cord, it's going to innervate multiple fibers, but usually they're all the same type. So you're not going to usually have type 1s and type 2 or type 2Xs all attached to the same nerve. Right? So you'd have all type 1s on that nerve. So that motor unit is all type 1s. Another motor unit might be all intermediate fibers, type 2As or 2Xs. But that doesn't mean the fibers are all clustered together. They're dispersed throughout your muscle. So if you think of the pool noodles, they're all different colors, but you're going to have blues on the outside of the pack, maybe midway, and then all at the center. Think of those as all type 1s. They might all have the same motor neuron, but they're obviously spread out throughout the muscle. So if you're thinking of the surface EMG, we're putting this over the muscle belly. We know that these are where our fibers are oriented. This is where we're going to put them. And if you have a motor unit that has fibers widely dispersed across that muscle, it's going to be harder to pick up the action or electrical potentials of muscle or fibers that are more deep within the muscle. Even though they're still attached to the same motor unit, they're attached to the same nerve, but if they're located more deep within the muscle, then that could affect your surface signal. So that's what it means by the spatial distribution of muscle fibers could affect that frequency signal. Electrode location is also a huge one. There are recommendations, like I mentioned before, that will tell you exactly how far down the muscle belly these things should be located. If there's a specific pennation angle of the muscle, you'll usually orient the electrodes according to the pennation angle so that you're along the lines of contraction when that contraction occurs. And there, you can find recommendations for almost every accessible muscle, whether it's in the face, hands, feet, torso, legs, arms. They have them for almost every peripherally accessible muscle. Some more deep muscles, like the one that was frustrated me, is the vastus intermedius, very tiny little muscle. Some people, you can access it and some can't. It depends on just their morphology and structure. Just some people are different. And there's a group out of Japan who's been trying really hard using MRI studies to get some hard set recommendation for the VI, but so far as I know, they haven't done it. My advisor was trying to push that on me and I sidestepped it. So there's some that are definitely easier to find than others. So the analysis of the signal. So we have our placement, we put our surface electrodes, we have our kind of raw signal. So how can you go ahead and analyze it? We're trying to derive this neural drive, right? So we're trying to tease apart the estimate of these electrical current changes across the muscle fiber, also referred to as muscle activation or neural drive. And we're looking at these discharges across the motor neurons. This is an example of a type of wavelet analysis. And you could see the different kind of spectrums of neural discharge according to potential different motor units that are being picked up, different fibers that are being picked up across the signal. So similar to if you have a prism and you're shining a light through that prism, you're going to get different bands of color coming out of it. It's the same concept where we take our raw EMG signal, that like green burst that you saw, we're going to put it through a type of calculus-derived spectrum or glass, and that's going to give us these different wavelengths. And these different wavelengths are different frequency wavelengths that can help us determine what type of muscle fibers or the fiber properties that this gross signal is picking up. So it's just another way to kind of tease it apart. This is not a super common way to analyze it using wavelets. It's more complicated. Most people will just use average root mean square, so looking at just the average of or standard deviation of that peak power of your amplitude, and then just hertz. So how many cycles per second for our frequency. But this is just another way to interpret that. So if you're trying to then interpret these signals, if we have an increase in our EMG amplitude, again, an increase in that root mean square, that will either occur when the discharge rates or the number of active motor units increases. Because again, remember, it could either be recruitment or firing rate. So recruitment means you're recruiting more muscle fibers. If you think of, if you're trying to do an exercise or a basic lifting task, if you were to go and pick up a box that was empty on the floor, you know it's empty, it doesn't weigh very much, you can pick it up, no problem, bring it up to, I guess, waist level and set it back down. Now, if you walked up to a same cardboard box and it was closed and someone told you there's 200 pounds of weight in that box, so you need to be really careful when you go to pick it up, when you go to bend the box, you're probably gonna watch your posture a little bit more, like psych yourself up, okay, get ready, this is gonna be really heavy. If you go to pick up that box thinking it's 200 pounds and it's not, it's empty, you might throw that box in the air because you've recruited more muscle fibers than you need for the task, right? Usually we're gonna modulate our activation, our central nervous system, at least voluntary activation, based on the demands of the task. So if it's an empty box and you know it's empty, then you would recruit your lowest threshold motor units, the smallest fibers, because you know you don't need a lot of force production capabilities. It's probably not going to be that taxing in terms of intensity, so you can just recruit those low-level motor units. If you think it's going to be a very difficult task, then you're going to recruit your larger, higher threshold motor units, more motor units even, because you know you need as much muscle as you can recruit to pick up this heavy load. EMG will try to help us figure out which one of those strategies is going on. Recruitment, meaning are you just activating a small number of motor units or a lot? Or firing rate, meaning that after you've recruited the motor units, you're sending that signal, and what creates force? What produces force when you're doing a muscular contraction? Well, that's true, everything is, so what parts of protein in your muscle? Myosin, actin, right? So myosin, actin, the myosin cross-bridge head cycling. So you're saying, okay, I need to pick up this box. Awesome, you pick up the box. But then if you still have to hold the box, then you're going to have to maintain the recruitment of those muscle fibers and maybe continue the frequency of the firing or the contraction of those muscle fibers as long as you're maintaining the task. So if EMG amplitude goes up, it could be because you recruited more overall muscle, or it could be because your central nervous system is saying, we're still maintaining this contraction, you need to continue to fire. If you increase that firing rate, you can increase the amount of torque or force produced from a single muscle fiber. So you can improve force or increase force by either recruitment or firing rate. So that's what we're trying to tease apart with the amplitude. Again, it's just an estimate, but it's a better estimate than not having anything.
You said those can increase work.
Yeah, so when you're trying to increase force production or torque production, if you're talking about like on a dynamometer or rotation around your joint angle, then your two ways of modulating more force is either you're recruiting more or you're recruiting what you have more frequently. So say you have a set amount of motor units in your muscle, right? So you're not going to be able to build a bigger VL while you're doing an exercise task, right? You're stuck with what you have. So you can recruit up to a point. When you can't recruit anymore, you can recruit them more frequently. So you can fire more frequently, send signals down the nervous system through that muscle fiber across that nerve more frequently, and that could give you a little bit more force production once you've maxed out recruitment. So usually you're going to recruit up until close to it's like 80% of max, and then you're going to start seeing more modulation or changes in the EMG signal as a result of firing rate. That's kind of like your last line of defense. Similar to if you ever do resistance training and you've done like barbell bench press versus dumbbell bench press, you've noticed that you can lift more sometimes if you do single dumbbells than the summation of the bar. Have you guys noticed that, or you've heard about that before? Right, okay. So it's similar with EMG, where if we look at the summation of our signal, which is what we're measuring, we're not gonna pick up everything that's going on. It's gonna be lower than if we looked at the exact signal of all the individual motor units. So this kind of answers your question, Zach. If you look at the bipolar surface electrode signal versus the intramuscular, the bipolar is always going to be lower. The summated signal is always going to be lower than the signal that would be produced from each individual motor unit itself. So we're losing some of that activity because we're kind of averaging it or summing it across multiple motor units. EMG amplitude is also a little bit less sensitive to low threshold motor units. And part of this is just because of how we analyze it and how we filter that signal. So we have the bipolar surface electrodes. So we have two, a positive and a negative electrode that are going to sit on top of the muscle belly. And we're going to filter out, in part, some environmental noise, but the rectification process of that signal to account for our action potential moving in separate directions. We're also going to lose a certain percent of our maximal power spectrum, or the peak of that signal graph. Our low threshold motor units fall below that threshold. So if we're filtering out everything that's less than 60% of our max power, our low threshold motor units fall below that 60% threshold. So we might not even be picking up what they're doing using surface EMG. So if you're recruiting a little bit of muscle to pick up an empty box, you're using your base level, low threshold, slow type motor units, the surface EMG might not pick up those signals, meaning that if you're doing a low level contraction versus a high level contraction, the EMG signal may not show any change because it's not showing the difference accounted for by those low level motor units. So its application is really best if we're using a higher intensity contraction, so a greater percent of, say, your maximal voluntary contraction, greater percent of your 1RM, or doing some kind of a fatiguing or longer duration task that will ultimately result in the usage of at least your intermediate and higher threshold motor units. But again, because you're losing some of that fine detail, because it is a summated signal and it's not directly being measured at the muscle belly site like the intramuscular would, you will lose some of that information from the low-level units. If you're only interested in low-threshold motor units, then the intramuscular might be a better way to go. I think I just said this. So what factors influence the EMG signal? I don't think you have this slide either, and I'd be happy to provide it for you so you don't have to worry about typing it out. But these are non-physiological factors influencing the EMG signal. There's a lot, right? So there's a lot of things that can affect the EMG signal. It's much harder to get really clean data using surface EMG, especially if you're doing anything outside of a laboratory setting, right? So most EMG studies are going to have a very high level of control. You're trying to get subject groups that are fairly consistent with training status, fairly consistent in terms of body weight and height, body fat percent, so we can get rid of some of these controllable factors. So the shape of the muscle and the associated connective tissue can affect our signal, the thickness of the subcutaneous tissue, which I've talked about already, the size and distribution of the motor unit territories, which is kind of that spatial distribution of tissues, or the pool noodle example I talked about, the number and distribution of fibers innervated by each motor unit, which kind of plays into the distribution, muscle fiber length, because that will reflect that action potential propagating down the fiber. So you might have a better estimate of surface EMG or muscle activation if you're using a longer muscle to use a double set of bipolar electrodes. So it might not just be over the muscle belly, you might have two separate locations on the same muscle to derive that signal. Electrode size, shape, and placement, which we have recommendations for that. Skin impedance, which I talked about. Muscle fiber shortening. What you'll notice, and I'm like a slight disagreement, but I also understand you won't see as many of these studies done in aerobic exercise, so less in cycling, even less in running, because you're going to have more rapid shortening of the muscle fiber, and you're going to have potential movement of the muscle relative to the electrodes. So you might have some muscle movement that's actually both separate from the skin movement, which can occur in really rapid contractions that you would see during running or some cycling exercise. Any cycling study that utilizes EMG or MMG will usually control for cadence to ensure that at least the rate of contraction is consistent across the entire exercise bout and across the subjects. And that will in part kind of account for the movement of the muscle. Crosstalk, as I mentioned that briefly, and that could be if you are having a signal that's being picked up from accessory muscles. That's not the exact muscle of interest. Usually more than a single muscle is participating in a contraction. And so if that electrode is in close proximity to an accessory muscle, you might pick up what's called crosstalk. Some additional factors that would be deemed physiological are the average conduction velocity could vary from fiber to fiber. That could also change as a function of the activity that's being conducted, hydration status, electrolyte status, all those things can affect conduction velocity, and EMG is not going to correct for those things. Synchronization is one that you'll see a lot in the literature, and that talks about if you have multiple motor units, each of those motor units has a separate nerve fiber that is innervating those muscle fibers that account for the motor unit. Synchronization means that if you have a synchronized signal basically, or a synchronized command from your central nervous system going to multiple separate motor units at the same time, they will cancel out. So a summated signal or a synchronous signal may actually indicate a larger global activation, but if it's synchronous, then that activation will show up as being lower than it actually is. Because just like if you're walking in sequence or you're walking in a line, like a single file line with a group of 20 people versus a group of 20 people, if you're following that group in the sand, like you're a tracker, right, you're gonna say, oh, there was one person one person walking in a line, so I know I'm not in danger because I'm only following one person. Versus if you're following a pack of 20, then you can tell all the different footprints. It's kind of the same thing with synchronization. So you're picking up one signal when really it could be multiple. And that could obviously affect your EMG interpretation. So let's actually look at what these signals look like and how they've been studied a little bit. So this is the EMG torque relationship, and this is what you ideally should see is that isometric torque. And a lot of these studies are going to look at isometric contractions because they're very easy to control, they're easy to grade. But if you look at percent of max, in this particular example, these individuals were starting at, say, 10% of max all the way to 100%. And then we normalized their EMG, so we have EMG amplitude and root mean square. As force increases, you should also see an increase in EMG amplitude, right? So that's a very stereotypical response that you're gonna see. But again, we don't know if it's because of recruitment or firing rate. So if you just look at the EMG amplitude, or even if you have the frequency also, you can't really tell which exact neural strategy is occurring because it's a global measure of activation, not if it's separate recruitment or firing rate. So how are you supposed to know what's going on, right? Is there any other better way? There is a slightly better alternative, or at least a combination, and that's to add mechanomyography to the electromyography component. So EMG is our electrical signal. MMG is going to be the mechanical counterpart of EMG. So where EMG is picking up action potentials, changes in electrical current going down the muscle fiber, MMG is picking up the sound of those oscillations. So when actin or when myosin is binding actin, it causes a sound, and MMG can pick that sound up, right? So it's going to pick up the actual fibers oscillating and the sound that's being derived from the cross-bridge cycling. And what's great about MMG is that the amplitude and the frequency actually help us tease apart those motor control strategies. So this is a raw MMG signal, so hopefully you can see it does look different than our raw EMG signal. If you don't remember what the EMG signal looks like, here it is for you again. So we have torque or force production. So if you're on the dynamometer in the back, you're producing force at a particular angle, so that's going to be torque. So we have someone, will say, okay, you're going to have a leg contraction and you're going to try to reach 50% and hold 50% of your max. So someone's going to kick out against that dynamometer lever arm to about what they think is 50%, and they're going to hold that for a particular period of time. So that's our torque at the top. EMG is going to be our electrical gross muscle activation of that same contraction. So as long as the torque is being produced, you're going to see a change in amplitude and a change in frequency as that contraction is maintained. Now, MMG is at the bottom here, and you can tell that it looks a little more boring, right? So not a whole lot going on with the MMG signal. You're going to see, at least for if you're holding an isometric contraction, you're going to see a large initiation at the beginning of contraction. Then you're going to see as contractions maintain, so this is a steady state isometric contraction, you're going to see the signal pretty much flatten out. And then when the contraction's released, then you'll see another little blip. Why do you think that, what can you guys think of, the signal kind of flattens out here in the middle? So we get a lot of noise, a lot of electrical potential here. This is the mechanical counterpart, and we don't really see anything here. And this is a maximal voluntary isometric contraction. Any guesses?
Yeah, the myosin arm, you don't gravity didn't do something.
They're just, yeah, they're holding on, right? So if you're not doing a dynamic contraction, they've attached and they've pulled, and now they're maintaining force, right? They're maintaining that connection. It's picking up the noise, right? The cycling of our actin and myosin or any lateral oscillations. If you're doing an MBIC, you're not oscillating anywhere, really, or very little. So you're going to see an initiation of a contraction, which is going to create a lot of noise because you're getting into position, and then you're going to see a noise cancellation or noise kind of even out. Then we see a little bit more noise at the end when you release, right? So when actin and myosin become separated, you detach those cross bridges, then we'll see another little blip. If you're doing a dynamic action, like cycling, you're gonna see a lot more noise, right? So this is much harder to deal with. And there's specific recommendations for how much of the signal you are selecting. It would match up with cycling cadence. Usually you need to know joint position too during the cadence, and you'd have to go through and select individual little bursts that represent the actual concentric portion of movement of that contraction. So a lot messier. You're gonna see MMG a lot more frequently in resistance training, single repetitions, or isometric muscle actions. So how does MMG differ? Well, we know it's the mechanical counterpart, so it's more of the movement or the mechanics within our muscle rather than the electrical activity, but we can use it hand in hand with EMG, which is really cool. What do these sensors look like? So I'll bring my MMG sensor. They cost like $2,000, so we're not gonna use them just because they're so sensitive. Like, I've broken them in labs, and then you have to send them to France, and it takes like a year to fix. So in our current economic situation, I am not gonna have us use these, but I will bring them so you can check them out. So MMG sensors of varying sizes are used for different uses. Larger sensors, which are basically a magnification of the sensor I'm gonna bring in, are used to measure vibration on planes, bridges, to see if they are structurally sound, because it's just picking up a vibration. So if you want to see that a bridge can sustain a certain wind miles per hour, same thing with plane winds, then you can put this type of small accelerometer on that surface. And we're doing basically the same thing with our muscle fibers. So this would be considered a small sensor. This portion here, that's the small little accelerometer. This tube, which is probably about this big, that is the amplifier, and this is what's actually going on the skin. So we'll usually place that small EMG sensor in between our bipolar EMG arrangement. So we have our two EMG sensors, and then right in the middle we'll put our MMG sensor. So it's picking up any kind of movement associated with that gross estimation of muscle activation for that EMG signal. Here are some other examples of MMG sensors. The one on the left is an older, maybe like 1999 version. So they've gotten smaller as time has gone on. This is just another example showing it in between that bipolar surface EMG arrangement. So let's tease apart those three different aspects of our MMG signal. So this might help you remember. The first part is called a gross lateral movement. And a good example is a bodybuilder, right? So a bodybuilder or fitness competition, when they're coming out on stage and they're getting ready to be from a relaxed position to a pose position, that big movement of muscle, that's the gross lateral movement that we see at the very initiation of our MMG signal. So if you remember back to that signal, there is that first blip, that is our gross lateral movement. So anytime if someone says, like right now as you're sitting in your chairs, like contract your quads. You go from relaxed, quick contraction, that would be your gross lateral movement. So if we had an MMG sensor on your leg, you would see that quick blip. If you maintain the contraction, then you would see it even out. The evening out are those smaller subsequent lateral oscillations, cause very little noise because they're occurring at what's called the resonant frequency of the muscle. So at any one time, you do have actin and myosin attached. That causes or that generates the resonant vibration frequency of the muscle. Just like if you were to put one of these sensors on a plane wing or on a bridge, even if there's no wind or no noise or no cars, it's gonna pick up a signal, right, at the resonant frequency that that structure is creating. Same thing with our MMG and our muscle. The very last little blip you'll see at the end of the signal, that's the dimensional changes of the active muscle fibers going back into place, or we would see those at the transition from a concentric to an eccentric muscle action. So as the muscle shortens and elongates, you're gonna see, you just, it's like, we'll just have this on the slide. You can laugh at this bodybuilder a while longer. It's so funny. So the dimensional changes of that, the shortening and lengthening, you'll pick up from the MMGs as well. So here's our gross lateral movement. So again, bodybuilder pose, have that massive quick contraction. We'll have that first blip. Then we have the smaller subsequent lateral oscillations at the resonant frequency of the muscle, and then we have a little dimensional change at the end as you release that contraction. So any change, shortening or lengthening of the muscle, you're gonna see little blips like that, which is why, and I'll kind of just skip over our bodybuilder again. If you look at a cycling signal, cycling is a series of concentric and eccentric actions, right? Shortening and lengthening. So we're seeing, this is like not at the beginning of the action. So we don't really can't differentiate any kind of gross lateral movement, but we see a lot of those dimensional change oscillations, which is basically what this is reflecting. We don't have any resonant frequency because you're always doing some kind of movement. So if you're cycling at 65 RPMs, you're constantly cycling through, and then you would be picking off, like if we were gonna analyze this, we would have a burst here. So this would be concentric action, eccentric, where that muscle is not being activated. Another concentric burst, not activated, concentric burst. Noise from the movement, concentric burst. Noise from the movement, concentric burst, right? So you'd have to know exactly where those, based on the cadence that they're actually exercising at, where those concentric bursts would occur in order to pull out that signal. So if we have both of these, EMG and MMG, it allows us to better interpret our motor control strategies because MMG amplitude is a reflection of recruitment. So we know that if MMG amplitude increases, it is because you are recruiting more muscle fibers, more motor units are being recruited. If we look at the frequency, that's a derivative of the firing rate. So if MMG frequency goes up, that's because you're modulating force because you're firing or sending voluntary activation signals more frequently. Remember, EMG amplitude could be either, right? So if we have EMG amplitude and we have MMG, now we can look at what MMG is doing and then determine what is causing that change in global muscle activation from the EMG, because now we can tease it apart using this mechanical counterpart tool. So let's look at a little bit more about how EMG and MMG have been utilized in our field. So we're gonna look at some, I picked out maybe, I think it's just one study for each of these areas, and most of these articles that I talked about, I added on Blackboard. So you should have them in the Module 2 selected readings. So if you wanna read more about them. So muscle force is graded through motor unit recruitment and or firing rate, and that's what we were talking about earlier. And so when we overlay our MMG signal with our EMG signal, we can further tease apart which one of those is actually occurring, which of the strategies is being used to modulate force. So in this first study, this was done in 2004, 7 men and 3 women participated in isometric leg extension. So they're using a dynamometer very similar to what we have here. And they had these individuals perform this leg extension, meaning they're kicking out and holding against a lever arm at 10, 20, 30, 40, 50, all the way up to 100% of peak torque on the calibrated dynamometer. So what they do first is they would have the subject sit down. They would have them kick out as hard and fast as they can. That would be, they would do that 3 times to get their MVIC, or their maximal voluntary isometric contraction. Then they would do this modulation. So they would have, usually on the screen, they would have a certain goal. So, okay, here's your 10%. I want you to push out against this lever arm until you get to that 10% line, and then hold. They would have another line for 20%, 30, 40, etc. So the person has some kind of a visual estimation of where they're trying to get to. They would kick out against that lever arm until they reach that estimation, similar to like a biofeedback, and then hold the contraction. So they did this for all 10 subjects. And what did they find? So this is our normalized isometric MMG amplitude. So if we look at our percent of maximal torque, you can see that a low percent of torque, we had, and this is, if you kind of average it out, we don't see a significant change in MMG amplitude. What does MMG amplitude represent? You guys remember? Recruitment, right? So MMG amplitude is recruitment. So anytime you see MMG RMS, remember root mean square is the square root of our power spectrum. It's the same, it means the same thing for MMG as it did for EMG. So RMS is going to be our measure of amplitude. So if we look here at the bottom of our graph, we can see maybe a little reduction, but kind of a general like evening out plateau at a low percent of torque. So not really any big change in MMG amplitude at low levels of force or torque. But as we increase our torque production, you see that there is a concurrent increase in MMG amplitude, right? So that's going to go up as well until we get to about 80%. At about 80%, there is a plateau in MMG amplitude, and then MMG amplitude actually starts to go down. So above 80%, we're going to see a decrease in MMG amplitude. Can anyone think of why? 80% is kind of a lot, right? 80% intensity is kind of a lot. Can anyone think of what might be happening if you're sustaining an 80% of your maximal intensity contraction, which would cause the mechanical counterpart, right? So this is picking up any kind of mechanical change within the muscle. Why might that signal go down past 80%? Are you de-recruiting muscle fibers? Because is it getting easier? Probably not, right? Because it's 80% and above, which is going to be harder. So we want to recruit as many as we can. So if we're not actually de-recruiting muscle fibers, why is our signal going down? Hey, what'd you say? Right. Well, if it's isometric, it's staying the same, right? So length is the same. What did you say? Well, at our higher intensity, you wouldn't necessarily be utilizing, but that would occur lower than 80 or 90%. Anybody else? So remember, think back to that MMG signal, right? So you have gross lateral movement, bodybuilder posing. Then you have resonant frequency, so not really a whole lot of signal because it's just kind of staying still. And then you have that little change at the end when you go from concentric to eccentric. So if we see a decrease in amplitude, a decrease in the mechanical component of our muscle, if you're holding an isometric contraction, I know like most people don't exercise on a dynamometer, but like in your mind, in your chair, you're all sitting, you have your legs free. Kick out and contract. So kick out and contract your thigh muscle and hold it. So try to contract your thigh as hard as you can and hold. Now, the longer you hold that, you're probably feeling kind of tired, right? Does your muscle feel like nice and relaxed and smooth, or does it feel kind of hard and stiff maybe? Probably kind of stiff, right? So the stiffer your muscle is, the less movement is occurring and the less sound it's going to produce. So as muscle stiffness increases, we say compliance goes down. So your muscle becomes less compliant, more stiff, not creating as much of those lateral oscillations. We'll see our MMG amplitude go down because it's not making as much noise. It's still contracting, it's still there, but because it's becoming less compliant, we're having changes in blood flow, maybe some blood flow restriction that's occurring from a sustained contraction. That's going to cause less room for those lateral oscillations because they're surrounded by capillaries, right? So if you think of muscle swelling that might be occurring, all those things would decrease the sound that muscle is making, and that would cause a decrease in our amplitude. And that's actually what we saw in this study. So we saw a decrease in compliance because of stiffness, or what we call a fusion of motor unit twitches. So that means if you are recruiting as many fibers as you can, you're recruiting, you're having firing rate go up. Can you write on this board? Or are you okay? Okay, so you have your 80% goal. You're recruiting. Now you've recruited all the muscle fibers you can, so now you're just going to fire more frequently. So you're firing more frequently until you can get up to this point. Up to a certain point, you have all the motor units that you have activated. You've maxed out recruitment and you've maxed out firing rate. So you're signaling as fast as you can and you've recruited as many as you can. And you'll see, similar to like tetanus, you're going to see a fusion, meaning that you're not going to get any new activation at that 80% or above mark. So you're maximally recruited, you can't have any more graded potentials from firing rate, you'll see a tetanus or fusion of that muscle fiber. So it's maximally contracted, it's fully like saturated in terms of signal. And same thing if you ever had similar to like a muscle cramp that you can't relax, that would be kind of a fusion or a tetanus. And that's what's kind of occurring above 80%. And these are, usually most of these are specific to either isometric or isokinetic muscle actions, because that's most often where you're going to see EMG and MMG utilized. So that's our MMG amplitude. What did frequency do? So here's our mean power frequency. So remember, MMG frequency is actually going to tell us the firing rate. So how frequently you're sending nervous impulses down that muscle fiber. So we see that it's kind of plateaued in a similar way at that low grade percent, right? So 10 to 40%, no real significant change in firing rate. If we look at about 40%, so we'll say this is about 40%. So between 40% and 75%, we see that MMG frequency goes up. What does MMG frequency represent again? Firing rate, right? So firing rate. So amplitude is recruitment, frequency is firing rate. So as force increases, as we're grading our force higher, we're going to see an increase in our firing rate up to about 75%. Then at 80%, I'm going to skip to my next slide because I think I messed up that one a little bit. At 80%, we see that line doesn't go down like we saw in our amplitude. It actually stays about the same or maybe goes up a little bit. So our firing rate actually continues to increase past 80%. Our recruitment, though, or our EMG amplitude or MMG amplitude seem to go down that signal. So we can at least be sure that past 80%, if we're grading or changing torque at all from that 85 to 100% range, any grading and torque that we see is most likely a result of firing rate rather than recruitment. So these are, again, individual contractions. So they did a contraction at 10%, held it, then relaxed. Did another contraction at 20%, relaxed, 30%. Took the values for all 10 subjects, plotted those values, those mean values, and this is what we got. So it's fairly clear from these two sets of graphs that there are definitely differences in how force is graded across the intensity spectrum, right, from 10 to 100%, and that you can partially tease apart where recruitment is occurring versus firing rate if you're using MMG. In mean power frequency, the units are in hertz, so cycles per second. So if we overlay our two signals or two components of our MMG signal, this is what we get. So we have our MMG amplitude and our MMG mean power frequency. So we can see that between about 40% and 75%, both recruitment and firing rate are going up. And then after about 80%, we'll see firing rate continues to at least stay the same, if not go up a little bit, whereas our MMG amplitude signal, whether it's because of de-recruitment or because of stiffness or changes in compliance, is going to go down. I'm not going to go over all of these studies because there's too many to spend time on, but I've provided some citations. Some of these articles are on Blackboard. Otherwise, these are some good citations or studies to take a look at if you're interested in motor control strategies and how strategies can be derived using EMG and MMG. The next component of this lecture is resistance training adaptations, and I'm gonna wait and finish that up on Wednesday. So we're gonna stop here for today. We'll finish up the rest of this lecture on Wednesday, and then we'll start in on the nervous system on Wednesday too. So start at 2:30, and I'll make sure to watch the clock better and give you guys a break on Wednesday.
Can you go back a couple slides?
Sure.